Ja comencem. Molt bé, doncs, moltes gràcies, Héctor, per tota la gestió, per fer possible aquests seminaris en format telemàtic. I gràcies a totes les persones que us heu connectat per seguir aquesta presentació. El seminari d'avui va a càrrec de la Irene Tornero Pinilla, que porta per títol Metacommunities and Biodiversity Patterns in Mediterranean Temporary Ponds. És un seminari que està centrat que va ser el tema de la seva tesi doctoral, que va dur a terme, que va defensar, crec, si no m'equivoco, el juny passat.
No, al març.
Al març, perdona.
Fa un any.
Fa un any ja. Doncs com passa el vent. El març passat, i és cert perquè va ser just abans del confinament, per tant va poder fer la defensa presencialment i el va dur a terme amb el grup en línia de recerca d'Aiguamolls, amb en Xavi Quintana, l'Estefani Gascon i en Dani Boix. Endavant, moltes gràcies, Irene, i encantats de tenir-te aquí.
Gràcies.
Quan vulguis.
Okay, so can you hear me? Yes. So good morning, I'm Irene Tornero and I'm going to present the results of my PhD thesis that was defended 1 year ago. It is entitled Metacommunities and Biodiversity Patterns in Mediterranean Temporary Ponds, the Role of Pond Size, Network Connectivity and Dispersal Mode. Ponds are considered as biodiversity hotspots since they support a wide range of rare and endangered plants and animals. Moreover, they provide several ecosystem services, such as acting as stepping stones for migration, dispersal, and genetic exchange of species, and thus playing a key role in landscape connectivity. This role at landscape level is clearly seen when we shift from considering a single pond to considering a network of ponds, which is how we usually find them. In this thesis, we specifically studied Mediterranean temporary ponds, which are protected under the European Commission's Habitat Directive, because they inhabit endemic species and which are in many cases also endangered. Furthermore, they harbor a very specific biota with adaptations to deal with variability and extremity of the environmental conditions, even surviving a dry phase. So in this thesis, we worked with the spatial scale, since the spatial scale of ecological data encompasses both the local level, in our case, the pond, and the regional level, which in our case would be the network spatial extent. So according to the theory of island biogeography, we can consider ponds as habitat islands, and therefore we could test the species-area relationship, which predicts a greater number of species in larger areas. At regional scale, changes in the network spatial extent could cause changes in species richness patterns. So, as we have already seen, connectivity helps maintain biodiversity and the ecosystem functioning. So when representing landscape networks, the graph theory has been frequently used because it provides a robust procedure for the quantification of the regional network structure and of local community isolation, and thus it is a good tool to represent metacommunities. Moreover, we can obtain information on the degree of isolation or centrality that a node has with respect to the whole network. Among the studies applying graph theory, most of them were conducted in dendritic networks, and they were theoretical studies, and the ones that were empirical, most of them were conducted in river networks, whereas there are only few studies in networks of lentic water bodies or using observational data. On the other hand, only few studies have tried to detect whether the spatial configuration of connections between communities, that is, the network structure, can be an important determinant of community structure. So, in community ecology, it is essential to identify the role played by both the environmental and the spatial processes on determining the community composition at local level. Thus, in the case of ponds, within the environmental factors which could be determinant for communities, some of them act at local level, for instance, macrophyte density, habitat area, or water characteristics, and other environmental factors act at regional level, for instance, geology or climate. On the other hand, some of the spatial factors which are considered important for communities are the distance to the nearest pond, the spatial extent of the pond network, or the connectivity within the network. In a study which included up to 158 datasets from independent studies and that analyzed the relative role of spatial and environmental factors on the community structure, almost 50% of the variation in community composition was explained by the combination of environmental and spatial variables. However, the greater influence of environmental processes with respect to spatial processes, or on the contrary, the greater influence of spatial versus environmental processes at determining the community composition can also vary with organisms' dispersal mode. In this sense, the same landscape can be differently perceived depending on the organism, and therefore depending on the organism's dispersal mode, which can finally have an effect on local diversity patterns. As I mentioned before, ponds are not usually found isolated in the landscape, but they are usually found in a pond network. Therefore, the organisms which interact with the ponds encompass a metacommunity, since a metacommunity has been defined as a set of local communities that are linked by dispersal of multiple potentially interacting species. And as I said, certain factors, such as the habitat heterogeneity, the connectivity, or the scale, can modulate the mechanisms on which metacommunity dynamics depend. These metacommunity dynamics have long been considered by many authors as excluding and independent dynamics, but over time, the perspective has changed, and it has been seen that they are acting at the same time, and depending on the situation, some dynamics dominate over the others. So the exposed theoretical framework allows us to think about several hypotheses. The first one, due to the existence of a positive relationship between pond size and species richness, we expected that with the increment in connectivity within a pond network, this relationship would become less pronounced. Secondly, since a negative relationship between spatial distance and community similarity has been described, we expected that if the spatial extent of the studied area increases, this relationship would become less pronounced. In this second hypothesis, taking the conceptual framework proposed by Heino and colleagues as the basic framework, we wanted to include in it the role of connectivity and organisms' dispersal mode. In Heino's proposal, it is shown the percentage of variation explained of community structure with spatial extent, considering scales that go from within a river to different biogeographical realms. And thus, in the smaller scales, so in the left part, a predominant role of mass effects is expected, which means that the percentage of community structure explained by pure spatial effects increases, and the percentage explained by environmental effects decreases. If the area increases, a major role of species sorting dynamics is expected, and therefore the role of environmental versus spatial factors would increase. And if we continue increasing the spatial extent, dispersal will finally be limited. So here we will focus on relatively small spatial extents, besides the fact that a study across biogeographical realms has been criticized. Therefore, at small spatial extents, we would expect that if network connectivity increases, spatial factors rather than environmental factors would predominate, since we would expect the predominance of mass effects or neutral models. Whereas in areas with less connectivity, environmental rather than spatial factors would dominate, since we would expect a predominance of species sorting dynamics. In relation to the organism's dispersal mode, we propose that organisms could be entered into this framework, and we would expect that passive dispersers would be more structured by spatial factors, while active dispersers would be more structured by environmental factors. In the third hypothesis, we suggest that if connectivity within a network increases, considering in this case greater connectivity as the greater centrality of a patch in relation to the whole network, the local diversity would increase, whereas in the most isolated patches of the network, local diversity would decrease. On the other hand, in the most central patches, their contribution to the regional diversity would decrease, while in the more isolated patches, their contribution to the regional diversity would increase. So the main objectives of this thesis are to test the effect of pond size on the composition and structure of the macrofaunal community, and to analyze if other environmental factors could modulate the effects of pond size on the community, to test the relative role of environmental versus spatial factors as drivers of community assembly, to propose a new framework linking distance decay patterns to different metacommunity dynamics, taking into account both the dispersal mode of the organisms and the spatial extent of the study site, and to examine how network structure and connectivity within the network determines patterns of local and regional biodiversity in metacommunities, taking also into account the potential role of local factors such as pond size, habitat quality, and environmental variability. The sites chosen to carry out this thesis were these four. Two of them are situated in the northeast of the Iberian Peninsula, another one in the southwest of Portugal, and the last one in the island of Sardinia in Italy. We looked for sites within protected natural areas in order to minimize possible anthropogenic disturbances. We looked for sites with different spatial scales, and ponds were chosen to include the broadest possible range of sizes within its site. And also within its site, we chose that there were at least 3 small ponds, 3 medium ponds, and 3 larger ponds. And the environmental variability among the sites was similar, and therefore they are comparable. In relation to sampling, we measured several environmental parameters in situ, and some others were analyzed from water samples, such as nutrients or planktonic chlorophyll A content. We also considered some habitat characteristics, such as Ethel's index, which is an habitat quality index. For the macrofauna sampling, since we had to sample ponds of different sizes, we performed a different number of deep net sweeps depending on pond size, but we processed the same effort unit. Then in the lab, individuals were sorted, counted, and identified to species level whenever possible. So now we move to chapter 1, entitled Pond Size Effect on Macrofauna Community Structure in a Highly Connected Pond Network, which was already published. According to the island biogeography theory, there is a positive relationship between the area and the number of species. In the literature, we can find that in permanent ponds, this relationship was not always found, and in temporary ponds, the results are contradictory, since some of them did find a positive relationship, but others didn't. Moreover, there are few studies working with different taxonomic groups at the same time. On the other hand, pond size effect can be modulated by other factors, such as water trophic state, which is important for the structure of macroinvertebrate assemblages, habitat structure that can induce changes in food availability and predation, and connectivity, which is determinant for the accessibility of habitats by a higher number of species. Thus, the objective of this chapter was to test the effect of pond size on the composition and structure of the macrofaunal community and to analyze if other environmental factors could modulate the effects of pond size on the community. The site chosen to conduct this study was Network 3, that encompasses 12 temporary ponds within the municipality of Vila Nova de Mil Fontes in Portugal. Pond size was calculated with the Google Maps tool and then checked in the field. Connectivity was calculated taking into account the geographic distance between the ponds and the species present absent in the ponds. Macrophyte biomass was obtained as a proxy for habitat structure, and trophic state was evaluated by means of fixed index, which takes into account chlorophyll A, oxygen saturation, total nitrogen, and total phosphorus. We calculated several community structure parameters that can be divided into three groups. The first one is diversity indices, so we calculated species richness, Shannon-Wiener index, the index of faunal originality. The second group was the phylogenetic indices. And the third group is the group related with body size diversity. We performed an NNBS, where the environmental variables were fitted to detect possible associations between the patterns of species composition and environmental variables. And we performed a series of linear models to study the response of our community structure parameters to pond size and to the environmental variables. Finally, we looked for possible covariation between any of the environmental variables and pond size. So here we see the results from the NMDS with the environmental fitting. In the upper part, we see ponds ordered by similarity in community composition, whereas in the lower part, species instead of ponds are represented. We see in bold that only pond size was significant to explain the ordination, and we see in gray color the rest of the variables that were not significant. You can see that the abundance of some taxa increased in larger ponds, such as Aexnamista, whereas the maximum abundance of other taxa, such as Culex taylori, was found in the smaller ponds. Moreover, the position of the other environmental variables indicates that there was no covariation with pond size. Here, we see the results obtained for the community structure parameters. Any of the three groups of indices had a significant relationship with pond size, despite when we first see the patterns, it seems that there is a positive relationship. Here, we see that no significant relationships were found between community structure parameters and connectivity, tricks, or macrophyte biomass. So, summarizing the results from this chapter, we did not find a strong effect on pond size on the macrofauna, since on one hand, we observed an effect of pond size on composition, but on the other hand, we did not find an effect on structural parameters. Moreover, we didn't observe neither an effect of the environmental variables on community structure. The possible reasons are that studies that did find that positive relationship included smaller water bodies than the ponds that we studied. Those studies did not remove the sampling effort effect. And in our study, maybe it was due to the high connectivity within the pond network, which is related with the short distances among ponds. So this possible effect on the distances among ponds and connectivity led us to consider both aspects in the next chapters. So moving to chapter 2, entitled Dispersal Mode and Spatial Extent Influence Distance Decay Patterns in Pond Metacommunities, which was already published. This chapter is framed within the theoretical framework of the distance decay of similarity, which is a tool to study the species assemblages responses to environmental and spatial variability. So distance decay is usually represented by showing a community similarity in response to an increment in distance among communities. So starting from this graphic representation, Heino proposed a model which linked the distance decay patterns to the main metacommunity dynamics. In relation to patch dynamics, Heino pointed out that it cannot be identified within the distance decay framework, but the species sorting, for the species sorting, a decay in community similarity with environmental distance would be observed. For the mass effects, a decay in similarity would be observed with both environmental and spatial distance. And finally, in neutral models, similarity would only significantly decrease with spatial distance. However, Heino's proposal did not take into account other aspects that I already mentioned, which can be determinant, such as the spatial extent of the studied area or the organism's dispersal mode. Therefore, in our proposal, we included those two aspects. And here you can see that we indicate the geographical distance with a continuous line and the environmental distance with a dashed line. We will compare the responses in two areas with different spatial extents. So we indicate in green color the small spatial extent and in purple the large extent network. Thus, we expect that the similarity decay in the larger network would be less pronounced for the three dynamics. Moreover, we think that we could also include organisms with different dispersal modes, such as the macrofaunal active dispersers, the macrofaunal passive dispersers, and the plants. And we think that they could be related with the dynamics as follows. Active dispersers would be more related with species sorting, whereas passive dispersers, either fauna or plants, would be more related to mass effects or neutral models. Therefore, we worked with two objectives in this chapter: to test the relative role of environmental versus spatial factors as drivers of community assembly, to propose a new framework linking distance decay patterns to different metacommunity dynamics, taking into account both the dispersal mode of the organisms and the spatial extent of the study site. So the study sites were network 3, again, which here is the small extent pond network, with a maximum distance between the ponds of 1.4 kilometers, and network 4, which is the large extent pond network, with a maximum distance between the ponds of 5.3 kilometers And so it is approximately four times larger than the other network. Despite one is larger than the other, we should consider both as having small spatial extents according to literature, but this is an advantage since it means a more realistic approach for studying metacommunities. We sampled the macrofauna and the plants of these ponds. For the data analysis, we obtained the community dissimilarity matrix applying the Jaccard dissimilarity to the presence-absence data. To obtain the geographic distance matrix, we applied the Euclidean distance to the UTM coordinates matrix. And finally, to obtain the environmental distance matrix, we firstly performed an analysis to get a subset of environmental variables mostly correlated with biotic groups, and then applied the Euclidean distance. To determine if there was covariation between the environmental and the spatial distances, we performed some mantle tests, and to analyze the distance decay patterns, we used exponential regression models. In relation to the result of the mantle test, when crossing the community dissimilarity matrix with the environmental distance matrix, we observed that in both the small and the large network, we found a significant relationship between active dispersers and environmental distance, and also for passive dispersers in the large network. But when crossing the matrices of the community dissimilarity with geographic distance, we observed no significant relationships, neither in the small nor in the large network. Regarding the results of the distance decay, we observed that active dispersers in the small network, so the green lines, are significantly related with both environmental, you can see here, and geographic distance, and so we could say that they are dominated by mass effects. But in the large network, so the purple line, the relationship is significant only with the environmental distance, and thus we could say that they are dominated by the species sorting. In relation to passive dispersers, they showed a significant decay in similarity with environmental distance in both the small and the large network, but no relationship with geographic distance. And finally, in the case of plants, a significant decay in similarity was observed only with environmental distance in the large network. So, to sum up, what have we seen till now? We have seen that geographic distance doesn't seem to have a great role in structuring communities, these communities, whereas environmental distance has a greater role. This would indicate that there is no dispersal limitation for the studied communities, with the exception of active dispersers in the small network. On the other hand, we observed that organisms with different dispersal modes can show similar responses to similar environmental gradients. It seems that there is no significant relationship between these communities and geographic distance, except for active dispersers in the small network. And when studying the effect of changing the spatial extent of the studied area in organisms with different dispersal modes, they presented different responses. So therefore, we could say that the metacommunity dynamics detected in each one network were different, since in the small network, passive dispersers seem to be driven by species sorting, active dispersers by the mass effects, and we did not detect a clear pattern for plants. But in the large network, however, the three dispersal groups seemed to be driven by species sorting. So to conclude, we could highlight that species sorting prevailed over other mechanisms. and that mass effects did not prevail despite we were studying small spatial extents. As we have just seen, connectivity is related to dispersal. That's why in this third chapter, we are going to see if connectivity can modulate biodiversity patterns. The chapter is entitled Disentangling the Effects of Network Connectivity on Biodiversity Patterns in Macroinvertebrate Metacommunity, which is now under review. So dispersal can play a double role since, on one hand, it can transfer the effects of regional heterogeneity to the local scale, and on the other hand, can lead to a progressive homogenization at the regional scale. Therefore, the exchange of individuals by dispersal is highly determined by landscape connectivity and is what finally determines the metacommunity functioning. Graph theory is a tool which is being used in metacommunity ecology since it provides a robust procedure for the quantification of the metacommunity network structure and local community isolation. But the number of empirical studies dealing with the role of habitat connectivity is quite low in comparison with the number of theoretical studies. Among theoretical studies, they have shown that the spatial arrangement of patches has considerable impact on the structure at both local and regional scales. From a graph analysis, we can obtain certain connectivity metrics which inform us of the gradient of centrality isolation of a patch within a network. So, according to literature, in dendritic networks, there is higher species richness in the more connected sites, and this pattern has also been described in some pond communities. According to previous studies, some habitat characteristics, such as habitat quality, habitat size, or water characteristics, can also modulate biodiversity at local and regional level. So here, as a quick reminder, remember that alpha diversity is the mean species diversity of a pond, in this case. Gamma diversity is the total species diversity in a landscape or region. And beta diversity is the contribution of a pond to the regional diversity. So based on previous studies, we are going to check if an increase in connectivity would generate a decrease in beta diversity, but an increase in alpha diversity. If according to one of the few works on pond networks, the environmental variables rather than spatial variables would play a greater role when explaining beta diversity. And if according to some studies with diverse scopes, when isolation increases, both taxonomic distinctness and average taxonomic distinctness decrease. Therefore, the objectives addressed in this chapter are to test the relative role of environmental versus spatial factors as drivers of community assembly, and to examine how the network structure and the connectivity within the network determines patterns of local and regional biodiversity in metacommunities, taking also into account the potential role of local factors such as pond size, habitat quality, and environmental variability. We calculated as network descriptors the linkage density, which is the number, the average number of realized links per water body, connectance, which is the number of links realized divided by the maximum possible number of links, and diameter, which is the number of steps required to move through the whole network. We also calculated some centrality metrics. So we calculated degree centrality, which is the number of direct links between water bodies, betweenness, which quantifies how frequently a water body is acting like a bridge to reach other water bodies, and closeness, which quantifies how close a water body is with respect to the rest of water bodies in the network. So these metrics reflect a gradient from a local or neighboring scale to a global or regional scale. We also calculated some biodiversity metrics. At local level, we calculated the ones already mentioned in the first chapter, so the ratified species richness, channel diversity, taxonomic distinctness, and average taxonomic distinctness. And at regional level, meaning that although we calculate one value per pond, they are metrics that take into account the rest of the ponds within a region. And they were LCVD, which is the local contribution to beta diversity, and SCVD, which is the species contribution to beta diversity. Once we obtained the SCVD, we calculate for one pond the proportion of species with an SCVD value higher than the mean of the ponds of the same network. Here you can see the studied networks. In this case, the networks include not only the sample ponds, but also the rest of water bodies of the region. As you can see, they are networks with pretty different structures. From the network 1, which is the biggest one and with a high linkage density, to the network 3, which is the smallest one and less dense, as indicated by connectance and linkage density values. And the other two, so network 2 and 4, which have in-between characteristics in relation to size and connectivity. Regarding the data analysis, first of all, to sum up the variability in water characteristics, we performed a principal component analysis, and we kept the first principal component to posteriorly use it as a variable. Thus, we wanted to relate centrality metrics, water characteristics, habitat condition, and pond size with biodiversity, and to do so, we performed a series of linear regression models for the local biodiversity metrics and a series of beta regression models for the regional biodiversity metrics. Then, following a model selection procedure, we obtained the best models for each metric. In this slide, I'm trying just to explain you how we graphically represented the best models. With a black dot, we represent the slope of the response variable. If the dot and its error bar are situated above the zero line, it indicates that a positive relationship exists between the explanatory variable and the response variable. But if the dot and the error bars are situated below the zero line, it means that the relationship is negative. And if the dot and the error bars are touching the zero line, it means that no relationship between the variables exists. So here I show you the results of the best models for the local biodiversity metrics. In general, among all the regressions, we see that there are some patterns more robust because the four networks presented the same response, whereas there are some cases in which the relationships between the variables depended on the studied network. Thus, we observed a robust and positive relationship between closeness, so the fact of being more central within the network, and channel diversity. We also observed relationships which are not so general and where being positive or negative depends on the network identity. So this is the case, for instance, of the relationship between species richness and closeness, or between water characteristics and species richness. We also found a relationship between pond size and taxonomic distinctness, which is positive or negative depending on the network, and something that also happened with the average taxonomic distinctness. Here you can see the results of the best models for the regional biodiversity metrics. In general, these relationships were more robust than the ones for local biodiversity, meaning that the four networks presented similar results. We observed a robust negative relationship for both closeness and ethels with LCBD, and a robust positive relationship with pond size and between pond size and LCBD. Moreover, we observed a robust positive relationship between ethels and SCBD, and not so robust between closeness and SCBD. So, to summarize, we observed the role of centrality metrics, but also of habitat characteristics on determining the biodiversity of the ponds at both local and regional scales. Specifically, we observed that patches with higher closeness presented higher species richness and higher channel diversity, and moreover, and as we also expected, the increase in closeness was related to a decrease in the local contribution to beta diversity and therefore to more homogeneous communities. In relation to the role of the habitat characteristics, we found that the ponds with the best habitat condition presented a higher percentage of species contributing to beta diversity, but at the same time, they are sites with lower contribution to beta diversity. This could seem contradictory, but it is not. And it could be explained because a site with a high LCBD value doesn't mean that this site harbors the rare species or that it has a lot of species, but on the contrary, it means that the site has a combination of species which is far from the main community. Moreover, in our case, since all the ponds had a good or very good habitat condition, those ponds that presented the worst conditions would be the ones with higher LCBD values. Regarding pond size, the results indicate that larger ponds could also contribute more to beta diversity, something that could be explained by the fact that as they are larger, they can probably harbor more unusual combination of species. And the relationship between pond size and the relatedness indices was not that robust, since in some cases, an increase in pond size favored a greater taxonomic distinctness, and in other cases, it was the opposite. Thus, as other studies have also pointed out, spatial factors such as connectivity and habitat characteristics act as drivers of biodiversity, both at local and regional scale. So now we move to the general discussion section, where I will try to integrate all the topics that we have seen till now. Regarding pond size, we have seen that neither in the smallest network nor in the rest of networks with different spatial scale extents, we didn't find the expected positive relationship between area and species richness. But we did find a positive pattern between pond size and the contribution to beta diversity. And we also found a relationship between pond size and community composition. In relation to the role of environmental versus spatial factors, according to previous studies, we expected in the smaller spatial extent network a greater environmental control than in the larger network, and in the larger network, therefore, we expected the spatial structuring, that spatial structuring would be stronger on the community composition. But we did not find a stronger environmental control in the smaller network in comparison with the larger network. And on the contrary, environmental control was more evident in the larger network. And finally, we observed a clear role of connectivity in structuring metacommunities in networks with different spatial extents. For instance, in the smallest network, connectivity seems to be responsible of the absence of relationship between area and species richness. And additionally, we got evidence of a strong relationship between connectivity and biodiversity. And as we expected, connectivity was positively related to alpha diversity and negatively related to beta diversity, generating then more homogeneous communities. And that's all. Thank you very much.
Thank you very much, Irene, for your presentation. I forgot to mention before that the questions must be written in the chat in YouTube, because we've moved the presentation from Meet to YouTube. So if you write down your questions in YouTube, then I will transfer it to Meet in order to be answered by Irene, so she can answer your questions. So I will do the same. So just from my side, I will write the questions and she can answer it. So not to do it in a different way from the rest of the audience. So we will change this again for the next seminar. So we think it's better to interact through Meet platform than in YouTube. It was just a try. So it's good to try new things and just to compare. So I will ask you to please write down your questions and doubts in the chat in YouTube, and then I will transfer to Irene so she can answer. So I have a question. So I just write you down a question. Then Irene, if you can ask, please.
Okay.
So
if I can see it.
Yeah, just a question. Just a second.
Okay, I have a question. The question from Frederick. I'm going to read it because I think that you cannot read it, of course. So it says, how does the dispersal depends on a single species in terms of being not active, but passive dispersion? So eggs, seeds. I'm not sure if I understand it completely, but
well, I can clarify if will be.
Yes, please.
My question is, so do you differentiate between active and passive dispersion, so that an organism is moving by itself from one lagoon to another, and passive dispersion would be when an organism is carried by another one?
That is the question. If the passive dispersal, the organism depends on another vector to be dispersed. It can be the wind, or it can be another animal, bigger animal probably. So birds or mammals even.
And there are...
But you're right that I think that now I understand that you mean that also these eggs, no, that can be passively dispersed.
Yes.
Yes.
Yes, my question was if not the living form of an organism is moved, but just the eggs or resistant forms.
Both, I think both. In fact, no, because also the adults can be passively dispersed by wind or by other vectors, animals. Okay. There's another question. It says, how do climate conditions determine the dispersal in the studied regions or in others? Well, that's a good question. I guess that, for instance, in places where there is a lot of wind or a directed wind, this is really important for the macroinvertebrates, in fact, because there are studies that have seen that predominant winds can direct to a specific area the macroinvertebrates. I mean, they can be really directed by wind. So their capacity can change, no? If they are active dispersers, if there is a strong wind, I think they might be conditioned by that wind, for instance, or by rain, I guess. But I was studying Mediterranean ponds, so all the regions are mostly dry regions. I mean that we haven't got lots of rains. Okay, the next question says, can these results be used for lagoon restoration in terms of predict how the diversity would change depending on the connectivity? Well, yes, I think, of course, you can use a similar approach as the one that we perform, because you can calculate the connectivity of your system, if it is a restoration system or a stored system, it doesn't matter, and you can calculate, for instance, as we did the local contribution to biodiversity, so it can give you an idea of how each of your lagoon is contributing to the regional diversity. I don't know if
Well, thank you, Irene. So my last question was concerning if well, you already answered if it would be interesting to consider the study for